Good morning, good afternoon, and good evening to everybody here and online around the world that is enjoying this Deep Sea live broadcast. I'm your host, Mike Irvin from the Fisheye Project.
And I'm Rachel Boshin. I'm a researcher here at the university looking at deep sea hydrothermal systems.
Yeah. So today, actually I'm really excited today, especially because this is our first live underwater broadcast of the Fisheye Project. So we're very fortunate to be a part of this event. We're here at the Ocean Networks Canada's Ocean Science Symposium with a live audience of students. And we're going to be showing you something absolutely amazing today. We're doing a ship-to-shore broadcast where we're going to be looking at deep-sea hydrothermal vents and some of the marine life that lives off of these areas and off these vents that are roughly about 2 kilometers underwater. So right now, off the coast of Fiji and Tonga, there is an expedition that's being led by Chuck Fisher of Pennsylvania State University, Peter Gerges of Harvard, Roxanne Beinart of Woods Hole Oceanographic Institute, and Vicky Farini of Columbia University. We will be speaking directly with Charisse Dupree on the research vessel Falkor, operated by the Schmidt Ocean Science Institute.
So before we get going, I thought I'd give you a little bit of a science background on what hydrothermal vents are. So we're going to be looking at these amazing structures underwater, and as a brief how they form, we have 2 plates in the Lau Basin. We have the tectonic plates you see across the world ocean. We have our Pacific plate, and we have an Australian plate. Pacific plate is going underneath the Australian plate. And as that happens, it melts, and you get these kind of magma chambers form. Cold seawater percolates down through the cracks, reaches your nice hot magma, superheated. As it comes back up through the rock, it dissolves bits of rock and it gets all acidic and it gets all hot and it gets all these minerals into the water. Then it gets to the cold seabed again, and much like if you let your coffee go cold with sugar in it, all of those minerals precipitate out and we get these gorgeous chimney structures forming. So what we're going to do now is we're going to...
I just want to do real quick.
Oh, okay, you want to do a quick plug?
So something that we want to let everybody online know is that if you have any questions during this experience, we encourage you to send us messages over Facebook at Fisheye Project, or you can also live tweet us. We have a social media team standing by to take your questions, and we will do our best to answer them as well. So if you can just get a quick look at this, we've got hashtag live dive, we've got hashtag ventlife_expedition, and you can tweet us at Fisheye Project, as well as at Schmidt Ocean. And we've also got at ropos_ROV. If you can add that in as well, that would be fantastic. All right, so
that leads us quite nicely into ropos. So I'm sure there are some blank faces going, what on earth is a ropos? Well, it's an ROV. It's a remotely operated vehicle which we can take to the depths of the ocean. So this particular one, it's Canadian owned and operated, and it can go to 5 kilometers. So the 2-kilometer dive we're doing today is well within our technological reach. Giant robot. It's attached by a big cable to the ship, and we can control it in real time. The pilots can decide where it goes, what it does, how it collects. It's very big. It's sort of 5 foot by 7 foot. It weighs something like, I think it's 6,000 pounds. It's a really massive beast of a machine, and we're very lucky to have it here to do our work for us today. Unfortunately, we can't control the weather in the tropics, and the weather last night was pretty bad. And this is a very heavy robot. It takes a lot to launch it, and the seas were doing this. So the robot's on its way down, but it won't reach the seabed in time for us to look at that footage live today. Yep. But instead, what we're going to do, we've got some footage from another dive. We're going to show you that and talk about that, and we're still going to be linking to Charisse live in the control van.
Yep. Awesome. Actually, do you want to pass it over to Charisse?
Oh, so now we can go to Charisse.
Yeah.
So Charisse, if you'd like to maybe introduce yourself, a little bit about the research you're doing, and also where you are exactly in the world right now and what the aim of the dive we're going to be showing is.
Okay, well, hi everyone out there. I'm coming to you live from the Falkor, and I'm sitting in the ROV control room right now with one of the Ropos pilots, Peter. Like Rachel said, Ropos isn't on the seafloor right now. We're actually on the deck trying to launch. The weather hasn't been wonderful, but we found a time where it's going to be safe to go in the water, so that's what we're doing right now. The cruise that we're on, like Rachel said, is a collaborative cruise between a bunch of different universities in the United States. We have a team of international scientists with us, and The objective of this is to study hydrothermal vents, so everything from the geology of the area on large scales to the fine scale spatial distribution of animals on and around the vents. We have molecular scientists on board that are going to do population studies. We have live animal studies going on board. And right now we're over Tahi Moana, which is a new site that was only just discovered in 2009, and we're going to be diving there today. We'll be diving to depths of 2.5 kilometers, which is about 1.5 miles. The video that you'll be watching, though, is a dive that we did a few days ago to a place called Abe, and it's about the same depth, so we go a couple kilometers down. That trip will take us anywhere from an hour and a half to 2.5 hours. So before we go to that, I'll just sum up by saying that, you know, there's a wide range of scientists on board, but the objective is all the same, and that's just to gain knowledge about this ecosystem that really was just discovered a few decades ago. You know, it's important for biodiversity, but it's also important because of the new anthropogenic impacts that we're going to be having on the ocean through things like deep sea mining. So that's what we're. Okay. Pass it back to land. You guys want to start the video?
Okay. So whilst we're starting the video, when I was talking about these massive hydrothermal vent structures, what we're really interested in isn't just because they're big and beautiful, and you'll get a really nice shot of them in just a moment. It's because of that vent fluid that's coming out. The minerals, we've also got hydrogen sulfide, and it's that the entire ecosystem is based on. It's based on chemosynthetic bacteria that form the basis of the food chain. No sunlight, it's very deep, it's very dark. The only reason we can see it is the lights we bring down ourselves. So we're going to put the video on, and we're going to let you enjoy it for a few moments and kind of situate yourself in silence, because it's quite an awesome view. And then Charisse and I are going to give you a little bit of a background about some of the animals down there.
All right, here we go. No, actually, I wouldn't do it right now. All right.
So Charisse and I will now start giving you a little bit of information about what you can see. Charisse, perhaps you might like to go first and describe the scene for people in terms of what animals we've got and the sort of structures we can see on the screen right now.
Okay, so we're flying into a place called Abe, and it was a long-term study site that was established over a decade ago. It's a fantastic example of a chimney complex. So you have clusters of all these chimneys in a single area. This one in particular, because of its dynamic structure, was nicknamed Hogwarts. And obviously the first thing that comes into sight would be the amazing structure, the relief. If you notice from the surrounding sea floor, which would be relatively barren, when you come close to these complexes, you see that everything's moving. What you thought was sediment or substrate is actually just layers upon layers of biomass, snails, mussels, shrimp, everything you might expect to see at an intertidal pool. We have the deep sea equivalent living down there solely off this energy that's being produced by the hydrothermal vents.
So I was just noticing when you were saying that, Charisse, that we've got some little squat lobsters swimming around. So you can see the little white crab type things that keep going past. They're kind of backpedaling because they can sense the ROV is close and they're trying to get away. And then in terms of the other animals that we've got there, on your sort of the bottom right as you look at the screen, we've got some of those squat lobsters on the bottom. Oh, the camera's panned around. We've got some mussels. That's what you see in the big clumps. And it looks like we might be going to be doing some sampling. So Charisse, could you tell us a little bit about the sort of sampling that they were going to be doing on this dive and why, and maybe the order of things they do, because it looks like we might be getting a temperature probe in a minute.
Okay, so what we've been doing when we go to these sites is we'll large scale map them first. We'll go in and we'll photograph everything at such high resolution that you can make out the individual species. We'll turn those into mosaics so that we can really get a feel for where the animals are distributed. And then we'll go in and we'll do a temperature and a chemical profile. So that's what you're watching now is Ropos uses its manipulative arms to pick up instruments like the probe. And we go around and we survey the entire area so that we can make a map that shows the temperature gradient, the chemical gradients, and then what we'll actually end off with doing on a sample dive like that is we'll collect some of those species, we'll put them in pressurized containers, we'll bring them to the surface, and we have a lab on board that can keep them alive. And that's how we'll kind of round off an entire dive sample, is then by having the animals on board, doing behavioral studies on them, physiological, and then we'll end up by doing molecular studies on them. So you're seeing us midway through a sampling protocol right now.
Awesome. I would actually just like to interject for a moment. Does anybody here and online have any questions currently with what you've seen and heard so far?
Don't be shy.
Yes. Awesome.
Okay, so the question was, how long does it take to survey your average size vent? I think Charisse would agree with me, that's a bit of a piece of string question. I mean, it can take hours to do everything you need to do in terms of the getting the depth of it, the topography, doing your video around it, and then getting all of your biological chemical samples. Sometimes it can take years and we still don't know exactly what's going on at a site. But an average dive can be, at a site will be a good few hours, maybe 6 hours max. Would you agree with that, Charisse?
Yes, and also I'd add that because we have so many different projects working towards this common goal of understanding, some of our dives have actually been multi-day dives, and that's one of the amazing things about using a robotic submersible, is that you don't have people that need to come back up to the surface to have meals. We can just leave that robot down on the sea floor. And I think our longest dive right now at a single site is 36 hours, and we'll just go continuously in shifts just to study one site.
Well, I have to say, we beat you, Charisse, on the previous cruise. So I was on board this ship last time, so just a few weeks ago, and we had the longest dive recorded for Ropos, which was, I think, 74 or 76 hours. So the only reason that we have to come up is because something breaks or one of the cameras stops or we get to so many samples we have to physically come up. The machine can stay down there almost indefinitely. So before we take more questions, it might be an idea to show you what we're actually looking at again on the screen. So we've zoomed in again a little bit and you can see some of the animals. We've got the big white crabs that look a bit like beach crabs. We've got the abithigrade crabs wandering around, waving their arms. We've got some hot vent fluid in the middle, and you can see that the snails have kind of clustered around them. And I brought some goodies with me today. So we actually have some samples of the vent snails, which I'll bring over to the lectern. So you can't see from the screen necessarily how big they are, but this one here, that is one snail. So that would struggle to fit in the palm of my hand. So these are really, really big snails. They're very cool. And they rely on the chemosynthetic bacteria to survive, so they don't live anywhere else. And this particular species you only get in the Lau Basin, so you only get it where these kind of vents occur. What else have we got?
Oh, actually.
Well, we also have some shrimp in there. So this is, again, from the previous trip. This is actually a shrimp trap. So I put down a trap with some bait in it and saw what shrimp came in to try, and they got stuck, couldn't get out, and then I got them. So this was just one shrimp trap, and this is chocker full of vent shrimp. These are an alvinicarid species, but we also get some smaller vent shrimp, some opipale. Yeah, give me the more shrimp. Give me the shrimp, Mike. Honestly, what kind of glamorous assistant are you?
There it is.
Shocking.
I gotta find it.
This is why you need a
Got
biologist.
so many options.
So these are.
Ooh, baby ones.
We got lots of little shrimp. So you get different species of shrimp at the vents. And we're interested in how genetically connected the different populations are in terms of mining disturbance and what that could mean in terms of habitat loss. So Charisse, would you like to add a little bit more on what you're seeing?
Sure. So The snails, very neat, are actually foundation species at these vents. Like you mentioned, everything's living off chemosynthetic bacteria, and these snails actually have them within their body cavity, and they're essentially farming their own food. So they're playing this dangerous game of they need to be by the vent water, but obviously they don't want to cook themselves. So they need to be by the vent water so that their bacteria, the little microbes inside of them, can survive, so that they can live off that symbiotic relationship. But of course, it's that fine line between surviving and being in the fire. The shrimp, which are actually a lot smaller than your foundation species, are and the crabs are part of the predators in the environment. And you know, sometimes we get distracted a little bit from our sampling work that we're supposed to be doing because it's so active down there, and we can't help but stop and watch the crabs hunting all the different species out there, and all piling on top of each other. We call it a little bit of a dog fight when that happens. Very entertaining animals, very active, not what you would expect from the usual deep sea environment.
So actually, we want to ask again if there are any questions. We would be pleased to take them right now, especially online.
Please don't be shy.
Yeah.
Not any? Oh,
Yes.
yep. Okay, so the question was, how fragile is the structure? That depends on where you are in the world, because the vents are made of slightly different minerals. But some of them, if you were to accidentally knock it with the ROV arm, they would completely crumble. And that's happened to us on dives before. Some of the bigger, older, more sort of solidified structures, if you ram it with the ROV, it's not going to fall over. So it really depends on the minerals. Yep.
If I can add to that, some of it can be very deceiving. We'll go up to a structure and it'll look completely solid, but these structures in particular are called like beehives. Rachel, you must have seen a few of them, and they look like they're solid, and you go touch it and it just poof, turns into sediment and floats away in the water. So when the minerals are precipitating out of the vents, it takes a while for them to solidify, and so it can seem like it's a solid structure, but really it hasn't taken that form yet.
So the younger chimneys tend to be the ones that are easier to accidentally knock over or damage, and the older chimneys are the ones that tend to be a little bit more stable. So do we have any more questions?
We actually have a question from online, and the question is, do these snails feed off of necrotic tissue?
That's a very good question. So as far as I know, and correct me if I'm wrong, Charisse, the snails we were looking at a moment ago, so the big ones that I've got here, and also these very cool hairy snails. Yes, they actually have hairy stuff on their shells. They're very cool. They're also down there as well. They are chemosynthetic, so they have bacteria, and that's how they get their nutrients. But they also have other snails down there, which are a bit like the whelks that you would get on the beach somewhere, and those guys are predatory. So I've got some of those here as well. So Charisse, have you got anything to add to? So the predatory snails, would eat necrotic tissue if you gave them a chance. If there was a dead animal down there, these guys will find it and quite happily snack on it. Anything else to add to that, Charisse?
I think you're spot on, and I think you have to be a little bit faster than a snail to get to that tissue down there, because those crabs move incredibly fast, and they'll just dogpile on anything that falls to the ground.
Yeah, so the crabs and the gallophaid little squat lobsters that you can see, and the shrimp, they're all going to go for that dead stuff. So yeah, and the snails do move deceptively fast, but not as fast as the crabs. Do we have any more questions?
Yes, we do actually. We have another question about how would sunlight affect these deep sea creatures? Does light affect these deep sea creatures?
So that's a very good question, and they have actually done studies at areas where they've done long-term deep sea research at vents to see if light impacted the shrimp populations, for example, because a lot of the shrimp don't have eyes, they have some sort of light sensory structure. So vents give off a slight glow in the infrared spectrum, and the shrimp can kind of detect this. But what they're interested in is if you're constantly shining a light on something that's not evolved to deal with light, do you somehow damage them? And that study seemed to think there were no issues, that it didn't cause them any long-term problems. I don't know if Charisse would like to add anything to that, the effect of light on these animals?
On board that are actively doing that research right now with snails that we've collected from these sites. So Rachel mentioned that it's being done on shrimp, and we have scientists on board doing it on snails. Stand by for results.
And what I can add to that is that the animals aren't used to being off the seabed, but they actually can survive on the surface in sort of ambient conditions for quite a long time. So I know that the current crews are keeping them alive for research, and I'm sure Charisse can tell us a little bit more about that at the moment. But on the cruise I was on, there was only me, and there were a lot of animals to process, so I was having to just put them in buckets of seawater in the fridge, and then take them out of the fridge when I was ready to deal with them. And the shrimp were quite happy, the snails were quite happy just hanging out in the fridge. So they can survive for long times out of their pressure and normal temperature range, but obviously not very happy about it. So it looks like we might have another question. Mike's wandered over.
Yes.
Yes.
We have another question about what is the size of the crabs that you're typically viewing here?
So there are no laser scale on the footage, Charisse, but how big are the Bithygraid crabs and the Paralomus crabs you've got down there at the moment?
Right. So the snails we kind of use as a scale. And so I can't actually right now, my feed's cut out, so I can't see the exact footage that you're looking at. But if you could use the live snail that you had as a standard, all the animals are sort of the size of a fist or smaller. And I can't really elaborate more than that. Sorry.
Yeah, I guess that's probably about right. I don't have a crab specimen with me today. I totally failed. I've got them back in the lab, but I've only got very small ones. They wouldn't be so great. I think the big Paralomus crabs look a little bit like spiders. They've got very long, spindly legs. We were getting individuals where the body was about 7 centimeters across, and then you've got the legs sticking out. So some of the crabs can be pretty big. We'll take another question in just a moment. What you're seeing on the screen at the moment, Charisse, is this some sort of water sample, or is it a temperature sample? I'm not quite sure what I'm looking at.
Right. It's very likely that it's just a temperature sample. That being said, on the exact same probe, we have a very neat instrument on board the remotely operated vehicle. It's called a mass spec, and it can measure the chemistry of the water across the entire range that we're interested in. So everything from oxygen to H2S. And so it's very likely that we were both taking temperature and doing a chemistry profile of the water at the same time.
Thank you. So what's the next question?
Yes.
That was an excellent question. So the question was, does the age of the vent affect the kind of species you see? The short answer is yes. The longer answer is the different animals will arrive as larvae most of the time, and then they take time to grow. So you get a certain element of succession according to which larvae arrive first and how quickly they can grow. And also depends on the size of the actual vent and what kind of chemicals are being emitted. But no two vent structures, no two chimneys will be the same. They will always have slightly different patterns of what lives where. The animals will be a slightly different age. I don't know if Charisse wants to add anything specifically about the Lau Basin succession.
Right. So we've actually done a fair bit of research on this in the Fisher lab, and it's very neat. So coming back here in 2009, we were able to then build a model of everything we had seen since 2005 and make predictions about where community succession goes. And now we've come back, 6 years later, and we've come back to the exact same study sites, and we see that the predictions were accurate, and that there is this pattern of succession. So not just what larvae arrive first and how the chemistry changes, but some animals have to arrive first and have to establish first to make a suitable living environment for animals.
I lost her for a little bit there.
Charisse, are you still there?
Here, yeah.
There we go.
Good. Sorry, we just lost your last sentence.
Okay, I was just, I guess that was a punchline, but what I was saying is the Fisher lab has actually predicted the succession of animals. And so we know that certain snails have to establish first to then allow for other animals to live there. So you do see the succession of snails first, and then predators come in afterwards, then other snails and other mussels, and then there's the same pattern on the way out. And that's all being established now. Predictions have been made, and now we've confirmed them coming out here again.
So thank you for that, Charisse. And what's also important to remember is that a lot of these animals can move. So the snails can actually move a little bit, and they can actually choose the sort of microhabitat that they live in according to the temperature and the chemical environment. So the patterns you see now won't necessarily be the patterns you see sort of, in a few weeks or months as the vents themselves change. So it looks like we've got another question.
Yeah, adding on to, especially talking about the temperature in the water there, there is a question about what is the temperature around the vent in the immediate vicinity, and then as you go further out?
So do you want to take that one, Charisse?
For sure. It's actually a remarkably dynamic environment when it comes to water temperature. So we've been using the probe on the sub to do profiles for the areas, and right at the vent itself, we've made readings between 300 and 400 degrees Celsius, which is 3 times the point of boiling. And then we move a centimeter away, or like literally just right there, and it drops down to ambient seawater, and suddenly you're in 2 degrees of water. And so that's really the fine line that I was speaking about earlier, about being close enough to the water to use that chemistry, but of course nothing that we know of can live in 300-400 degrees of water.
So I know we're running slightly short on time, so very quickly, thank you for that, Charisse. The reason why 300, 400 degrees of water doesn't form steam, and that's probably a question on most people's lips, and that's because of the pressure. Sorry, it wasn't actually me asking you. That's because of the pressure. It physically can't form steam because there's so much water on top of it, squishing it down. And to illustrate that, I've got some polystyrene cups. This is what scientists like to do when they're on ship and they go a little bit loopy after 2 weeks at sea. So this is what a polystyrene cup should look like, and this is what it looks like after being sort of just over 1 kilometer down in the seabed. So this is what happens to your air pockets. They shrink, which is why scuba diving down there is really not a good idea. But this is the effect of pressure, and that's why the water doesn't actually boil.
Actually, we do have another question as well, and it's kind of looking at why do some of the animals here have color, especially at depths like this where the sun does not reach?
That's a very good question, and you'll notice that a lot of the animals don't have color. So a lot of the animals are white. That's because, say, there's no light, so there's no point in investing in developing color pigment. But some of the other animals, generally deep sea animals, will be pink or red. And that's because red light is the first light to kind of be lost in water. So if you're pink or red, that makes you invisible, kind of gives you like an invisible cloak, as it were. But a lot of the animals here are black or brown, and some of that isn't actually their natural pigment. So for example, the big snails that I've got here, all of the brown on the outside of that is actually a mineral deposit. That's actually iron oxide from the vents themselves. So there's so much precipitated stuff being thrown out of these vents, it's got to settle on something, and it quite often settles on the snails. So the snails can be black, they can be brown according to the minerals. Some of them, like the hairy snails I've got here, are white because that's a little bit of bacterial mat that's settled on them. I think that probably answers the color question.
Yeah, let's also go a little bit deeper into this as well. So how do these species actually navigate this kind of fine line between fire and freezing, crushing cold?
That's a very good question. It's probably best for Charisse, because I think you're doing work on that on board, am I right?
Yes, it's a different department than me. It's the people that deal with the small interior of the animals. So not sure I could field that one sufficiently. Rachel, if you know anything you can add.
Okay, so when you're talking about navigating that fine line, do you mean as in how they actually move around in the environment? Is that the question? Yeah. So bear in mind it's dark. So if we were to turn all the lights off in here and you would somehow have to find the food that's out there where all the lunch is, you might struggle a little bit. So, well, I don't know, you might be able to follow your nose, who knows? Maybe if you're hungry enough, you'll find it.
And actually, to add more onto that too, it's you have no light, zero light.
There is nothing there. It is completely dark, and that's why most of the animals don't have eyes, because there's no point. They've lost them over time. But some of the animals, like the crabs, for example, not the ones you can see in the frame here, but some of the bigger paralomus crabs, they have hairs all along their body, and that helps them feel their way. So like if they were to do this and they'd be like, what's that? Is that a crab? Is it a shrimp? Can I eat it? Do I want to run away from it because it's a really big crab? So they can do a certain amount of feeling, and they can feel if the water around them moves as well. So those hairs are great. And some of the snails have hairs. Not entirely sure why they have hairs. Maybe someone on board would have a better idea of that because they're working on the snails.
Or anybody online.
That's true. Maybe it's a sensory thing, or it might allow them to trap bacteria. Don't know. Charisse, do you know why are the snails hairy?
Right. Again, I don't know why the snails are hairy, but I can add to what you were saying. So sorry, I thought it was more a protein question and an internal. But I will say that what we've observed is when we watch these animals, it's not that they're making a mistake, but the very mobile animals will actually walk up to a vent and then literally jump backwards. So I think a fair bit of it actually might be trial and error, and that they walk and they actually wait till they hit that temperature gradient to then move a different direction and choose a smarter path maybe to their food source.
That's quite funny to watch as well, particularly if you've been watching this for many hours on board a ship. The animals can turn out to be a little bit funnier than maybe they actually are. The snails in particular are great because they'll end up in a really hot bit and then they'll try and sort of tumble over each other to get back to where they want to be. So we've got time for one last question, which I've got here. And the question is there great variation in water movement around the vents? So I guess in terms of current. I don't know what the currents are like at these particular vent systems. Charisse, can you add to that?
Actually, it's incredibly neat. So I think the driving force at an active vent is so much about the venting itself that you don't notice any animal distribution patterns because of current. But these vents also turn off, and we've gone to sites that don't have venting anymore, but they have the structures. And that's when you really see animal distribution and the effect of currents. So we'll have large brittle stars and basket stars climbing to these top of these now dead vents and utilizing the current down there. So there actually is a lot of variation and enough to then sustain ambient sea life in the areas once the venting turns off. So that's just evidence of the dynamic currents that you find down there.
That's absolutely right, and that can sometimes be that one side of a chimney you'll get nothing, so a dead chimney, so not like these, as Charisse said, you'll get absolutely nothing living there, and then on the other side you can have corals and basket stars because they're eating what's being washed off the vents themselves. So you have a halo effect. So I think, Mike...
We have a lot of sponges down here too, a lot of carnivorous sponges, and they're a great indication of where you're getting current and where you're not.
That's right. So I think it looks like we're running short of time, so Mike might have to do the wrap-up.
All right, well, I just wanted to actually say thank you very much for everybody here for joining us and everybody online. We do truly appreciate the involvement and the questions. This was, like I said before, our first broadcast working with everybody. So it's been fantastic on our end for the Fisheye Project side. We want to give a special thanks as well to Charisse and Peter that are in the control booth that we were just speaking with, and as well as Rachel, of course. And we also want to thank the Ocean Networks Canada and UVic audiovisual team. They've been fantastic in helping with this broadcast as well. We also want to give you a chance too, is that if we have not answered any questions, please retweet us. We're going to do our very best to respond to them. It might take us a little bit of time, but we're going to be doing that throughout the rest of the week here, actually going into next week. So the other part I wanted to talk about too, and this is just a last thing, is that Fisheye Project on June 8th, we're actually throwing on a special event, and it's a underwater broadcast off of Ogden Point, and we're looking at the kelp forest there. And this is going to be in giant screen theaters, as well as it can work on your phone, your touch tablet, your computer. It'll be in IMAX Victoria, and it'll actually be into a lot of other spaces in North America. So it's an opportunity that you can actually be a part of an experience similar to this, where you can actually see that unseen, and you can actually ask us questions and go diving without getting wet. So that's going to be June 8th. We're going to be plugging a lot more information about it coming over this period of May. So we'd be very happy to have everybody here be a part of that event. There's going to be a physical space as well at the Royal BC Museum. So please check out fisheyeproject.org if you'd like any more information. And thank you all very much for joining us today.